Hi everyone. Thank you for joining us today on this week's Exploration Club. So as my colleague has sort of noted, the spatial sort of the talk for today will be focusing on spatial transcriptomics and how this sort of data reveals distinct roles of SPP1 and HLA-DQA1 macrophages. So that will be sort of the focus of today. Sort of in the past sessions or the previous discussions, if you're new to the Exploration Club, we looked at data which established the heterogeneity of macrophages and their potential roles in lung cancer. Today, we'll delve deeper into sort of that knowledge and into the functional differences between SPP1 high and HLA-DQA1 high macrophages and how sort of this data can provide new insights into tumor progression. Okay. Okay, so a bit of a recap from the previous session, just so you're able to follow how this data sort of builds into this week's session. So in the previous sort of analysis, we explored the different macrophage subtypes using the meta-reference cell typing prediction tool from the bBrowserX system. After finding how SPP1 and FAP4 macrophages, we also discovered that there is an expression pattern of these macrophages where the SPP1 is sort of increased in lung-related diseases. And we see that FAP4 is decreased in this lung diseases. So they're sort of more highly expressed in the healthy donor samples. To further validate the finding, we explored a bigger sort of picture in this analysis, sort of using a specific lung atlas that we built from the Talk2 data system. And we also looked into a very specific spatial transcriptomics data set to examine the sort of spatial distribution of these three macrophages or these macrophages that we identified. Okay. So going more in the detail of how we were able to examine this using the cell typing tool, while examining the SPP1 macrophages using sort of the cell type prediction result, we found several other lung cancer research groups that annotated these clusters or identified the clusters as tumor-associated macrophages. So this opened up a lot of sort of analysis relating to the ability of SPP1 macrophages and how this relates to sort of tissue infiltrating macrophages. So you can see here on my sort of slide that we identified these two studies from the meta-reference tool. And this sort of also gives us detail into how the authors or their core markers in identifying this population. Okay. So if we go sort of deeper into sort of the CosMx data set that we used in the previous session, when we examine the CosMx spatial transcriptomics data set, we confirmed that the SPP1 macrophages were truly related to the tumor microenvironment. So from these figures that I'm showing here, and I'll turn on sort of my point so you're able to see it clear. So these sort of images that you're seeing here, these sort of figures showed us or confirmed that SPP1s are truly related to sort of the tissue microenvironment. And we can see specifically that these green macrophages or the region of these green sort of macrophages wrap around sort of the brown tissue of the tumor. And however, when we're looking at the SPP1 sort of transcript target in that case on the tissue, it's overlaid across all the tissue in the lung. So it could be expressed in the normal adjacent tissue within these samples and not only the tumor microenvironment. So this is something that we really want to understand and get further insights from other data and also looking more sort of deep into that sort of study. So if we go sort of into more like what these sort of results mean, so interestingly, we observed an inverse correlation between the SPP1 and the HLA-DQA1 expression. So while the HLA-DQA1 transcript is known for sort of its antigen presentation, the function of the SPP1 high macrophages remains unclear. So we're able to see that as well within this data set and the overlay transcript information. Okay. So I'll go into here. So to further sort of understand these functional differences between the SPP1 high and the HLA-DQA1, High macrophages, we compared their sort of gene expression profiles using the spatial transcriptomics data set. So the main sort of emphasis of this week is what are the roles of these sort of gene targets or these transcripts that are sort of enriched in these macrophages and specifically in lung cancer. So not across different respiratory diseases. Okay. So I'll go into more here, sort of the step-by-step process. So in order to sort of build further into this analysis, we'll sort of use the Cosmics data set once again to get further insights. So the first thing that we're going to do is sort of subcluster the macrophage sort of label from the niche metadata of the study. So when we select the macrophages, you can see directly it's sort of labeled the cells and how it's overlaid in sort of the brown tissue of the tumor. We'll subcluster these so that it isolates any other population that we're not really interested in visualizing and analyzing. And from here, we're going to try to identify both the positive or highly enriched HLA-DQA1 transcript expressed cells and also the SPP1. So in order to do this, I'll sort of try to sort of search for that specific transcript, A1. Okay, I'll click on sort of the expression. So in order to be able to really capture all of the different cells that are expressing that transcript, instead of selecting or gating only sort of a level or normalized level of that expression, what we're going to do is change the unit from a normalized to a binary unit. So I want to really capture all those cells that are expressing that transcript at some level. So in order to do that, I'll select the express label and I'll go on to sort of labeling these cells. So these would be sort of my tumor associated macrophage group. Okay, and I'll label these as HLA-DQA1 highly enriched. Okay. So once I add that label, you'll see that it automatically creates a label. And then I have this unassigned group, which are all the cells that are not expressing that transcript. So the next step is identifying the SPP1, which sort of is not concentrated on a very specific region in the tumor. So when I look for the SPP1 transcript, I can then do the same sort of process and sort of select to label sort of the expressed cells. So once I select the cells, I'll go to label. I'll append this existing group to the group that I want to append sort of the other HLA. So I'll integrate the two labels. And from here, I'll create one specific label for SPP1. Okay. So once we add these groups, I can then remove any of the unassigned, which are all those other macrophages that are not expressing those targets. So I'll select these two sort of groups and I'll subcluster those groups once again. Okay, so in order to really go into sort of what we're trying to analyze, so this was sort of the process in labeling the groups. And then the next step is what is the difference in the gene expression of SPP1 and HLA-DQA1 macrophages in lung cancer. So in order to sort of answer that, once our sort of regions or our cells are labeled, we can then go into the differential expression analysis, and we'll go into this pairwise comparison. So again, you'll need to sort of add a label to the analysis that you're performing. So TAM, HLA-DQA1. Okay. And I'll go into the group annotation. So I specifically want to select my TAM group labels with the sort of high characterization that I identified, and then I'll generate the analysis from here. Okay, so while this loads, one of sort of the differences or the results that we'll be able to capture. So we can see here in sort of the volcano plot, once you run the differential pairwise comparison, that we can see specifically that the SPP1 high macrophages exhibited a gene expression, sort of specific gene expression signatures that are associated with sort of the tumor metastasis and immunosuppression. So these sort of key genes, the FAP4, so the ones that I highlighted here, the SRGN, MMP12, and MMP9. These are sort of implicated in tumor progression and immune invasion. So how can I show you sort of these correlations with other data, not only single cell RNA-seq, but other sort of either immunostaining or other data that has supported these results. So if we go into some of the results or this specific study, the FAP5 gene here has been shown to regulate lung tumor metastasis through controlling sort of NK cell maturation. So this is sort of one of the insights that we can gather and how this relates to sort of the question that we were trying to answer. If we look into sort of the other sort of genes that we identified, so the MMP9 and the MMP12, so sort of these genes, along with the SPP1 enriched population itself, have been suggested to be markers of IPF transition in lung cancer. So via sort of activating that sort of pro-tumor inflamed macrophages in these patients. So this is another insight that we can gather even just from looking at the CosMx data set and how this relates to sort of the functional role of these key regulatory genes. So if we go into this sort of final other sort of marker that we see that has a potential in this sort of cluster, this SRGN sort of target has been shown to play a role in the tumor stromal sort of interaction and how it's able to reprogram sort of the aggressive immunosuppressive tumor microenvironment. And this has been specifically shown in lung cancer as well. So these are sort of other insights that we can gather and how this has sort of a pivotal role in the interaction of the tumor stromal environment. Okay. So if we go sort of into the next sort of question that we want to answer from here, once we sort of identified how this fits into their role and how this has other clinical sort of implications, we want to gather the next steps, some insights from the enriched biological pathways in these SPP1 macrophages that show to have some sort of relation from these other targets. So this would be sort of the next step. So being able to perform pathway analysis from the differential gene expression sort of results. So if we explore this sort of analysis that I've already shown, sort of the volcano plot and sort of the genes that are differentially expressed, we can go into the pathway analysis feature from here. And this will show us sort of some information relating to how the upregulation of that metastasis or the immunosuppressive genes play a pivotal role in these SPP1 macrophages and specifically in promoting lung cancer, since we are using a CosMx lung cancer data set. So we want to sort of gather further insights into these pathways. So if I go into sort of the functionality of the sort of pathway analysis in the DGE feature, we get some more information into sort of the different pathways that have a functional role. So in SPP1 macrophages, we can see that there's an enrichment of lung fibrosis as a pathway, and this may relate to the SPP1, the MMP9, and the MMP12, being sort of some form of prognostic markers for these lung cancers in IPF patients. So if we further sort of go into further analysis from here, we can see that the P13K and the mTOR pathway is significantly enriched in these SPP1 sort of macrophages as well. So the pathway has been considered as one of the most altered molecular pathways in lung cancer malignancy, which you can see from the result here and how I'm able to fit that to other translational oncology data that has shown the same sort of results as well. Okay. So if we then want to sort of further gather this information and understand other types of findings, so how can we further investigate SPP1 and HLA-DQA1 macrophages and how they're different across different types of lung cancers? So again, there's many different forms or lung subtypes, lung cancer subtypes that we want to really see if there's any other implications that we can gather from this. So in order to address this limitation, we can perform further investigation across different types of lung cancers. And we can use this by creating a custom data set that integrates over sort of 146,000 of cells. So in order to be able to do that, we'll go back into the system where we can see sort of the talk to data system here. Okay. We'll go into the talk to data and we'll try to sort of filter these studies here. So in order to go into this, we'll go into the talk to data system, the single cell RNA-seq in human. We'll go into the filtering tabs where we can select both the condition. So I'll select lung cancer here. Just so I'm able to identify all those different data sets. So I'll select all of the cancer, sort of lung cancer subtypes under the respiratory disease system. From here, I'll only want to concentrate on macrophages as well. So I have to make sure that I also sort of add a filtering for these specific macrophages. And the last step, just from filtering on the categories, is to also make sure that I'm selecting only cells expressing these particular markers that I have found some functional roles in this analysis. So I'll select the SPP1 as well as the HLA-DQA1 in this analysis here, just so I'm able to make sure that I'm filtering only those cells relating to that analysis. So again, I can see here once I create the custom data set that I identify these 146,000 macrophage cells, specifically having that sort of environment sort of expression pattern. And from here, the next step is to create a custom data set. So you can integrate all of these different macrophages that are expressing these particular markers in order to really increase the statistical power when we're trying to understand and sort of focus on these specific cells. So once that sort of custom data set has been generated, again, you can sort of generate it by creating it and adding sort of the label. And we'll go back into sort of what we can see from here. So the first analysis that we can see when we're sort of creating specific markers and we're generating different visualization tools, as well as being able to map other metadata associated with those studies, we can compare that the SPP1 and the HLA-DQA1 expression across different sort of tissue samples are relevant to the distinct spatial distribution patterns as well. So it is clear from these different visualizations that the SPP1 has a really sort of low expression in the adjacent normal tissue. So we can see that based on sort of the group here. And on the other hand, when we're looking at sort of the metastasis, we can see a high expression on the metastasis site relating to the SPP1 expression. So this is sort of some information we can gather. If we then look into the HLA-DQA1 role in this analysis, we can see that the normal tissue showed a high expression of this HLA-DQA1, which suggests that this sort of has a role, a protective role of this gene. So we can see then again, sort of this adjacent normal tissue and how HLA-DQA1 sort of has that specific role in the analysis. Okay, so from here, if we further sort of want to observe the changes in these macrophage populations following the treatment, and specifically looking at SPP1 sort of expression and how it sort of relates into that analysis, when we perform sort of the bar plots, we can also sort of split it by treatment time points as well to see if there's any indication that these specific macrophage or enriched populations has a role in the sort of post-treatment response. So we can specifically see that the SPP1 expression has decreased as well. And the HLA-DQA1 increased in this population. So this suggests that the HLA-ADQ1 macrophages has a sort of role in the response to the therapy from these different sort of lung cancers. So this is sort of some insights you can also gather when you're inputting other information relating to the time point, post-treatment, and also the type of therapies that are being used. So what can we gather from all these different results? So inputting sort of an atlas analysis, so where we increase the statistical power of this result, as well as the spatial transcriptomics sort of insights that we gather with other published data. So some of the main summary points from here is that the SPP1 macrophages can play a sort of significant role in suppressing the immune response against different forms or different subtypes of lung cancer. And it promotes metastasis as well. So we had a specific way where we correlated sort of the distinct spatial distribution patterns using different sampling sites from the data. Another sort of result that we gathered as well was that HLA-DQA1 as sort of a marker in macrophages promote immune surveillance, and it increases after cancer treatment. So we can see its relation to post-treatment, and we can also gather more information if we went even deeper into the specific time points of that therapy. So these are some of the main insights we gathered today. And I guess this sort of concludes my sort of presentation. So I'll go forward into allowing sort of the scientists in the group to ask specific sort of questions in the Q&A section. And my colleague as well will be sending sort of the survey, so you can give us some insights or some feedback as to what type of story you would like us to show as well. So not only in lung cancer or in the respiratory system, but it could be in solid tumors or other sort of an analysis that you would like to see. Okay. So I'll go into sort of the Q&A section now just to see if there's any questions in the chat. So I can see that there's already some questions that are open. One from Jasper. So how is this subset of SPP1 macrophages only specific for lung cancer? So can we see this in other cancer types? So yes, Jasper, if we answer this question, so yes, we do see it in other cancer types. So our team has been able to analyze across different types of sort of atlases that we generated from the system. And we can see that if you explore talk to data and you generate these different atlases, we do see the same SPP1 macrophage role in sort of the colorectal cancer and the head neck cancer as well. So this seems to mainly relate in cancers of the solid tumor when we've been analyzing across other cancers. Okay, so another question from Anya is, can we show how can you sort of access a Cosmics data set so you're able to show that? So yes, we can show how to access that. So in order to access any publicly available data that we curate in the Talk to Data repository, you have to go and explore the Talk to Data system. And when you explore the system, you have to choose what species you're focusing on. So whether it's human data, mouse experimental models, or non-human primate data as well. And you can select here the different databases relating to the different technologies that you're focusing on. So for any Cosmics or 10x Xenium spatial single cell resolution data, you would have to select that specific database. Once you have the database selected, there's different types of sort of questions you can answer from the system, whether it's gene focused, cell type, signature gene focused, or looking at just the studies that are curated in the system. So you can go into the studies tab, and this is where you can filter based on the specific tissue and see all of the different data sets that we have relating to lung cancer. So you'll be able to explore each specific study individually when you're sort of opening and exploring that study. Another thing I'll mention is that if you don't find many studies in your specific tissue that you're focusing on, you can also request data sets to us. So this is actually open for bulk RNA-seq data, for Visium HD or new Visium data sets that you want us to curate, or single cell RNA-seq as well. Okay. Okay. So is there any other questions in the chat that you need sort of some clarification on? Okay. So I believe my colleagues have also answered some of the other questions in the chat. Okay. So I guess in the next session, I'll give some time so that you're able to sort of answer the survey. So it's really important to us to see like if it's useful for you, or if there's any case study that would be more relevant to your analysis that you would like to see how we can use the system or different applications in the system. Please let us know, because we do have other different applications we haven't already sort of gone through in the Exploration Club. So please give us some insights into that. One last thing I'll mention is that this club, for anyone who is new to the Exploration Club and didn't join the previous sessions, this is a weekly session. So some of the sessions will be led by me as a host on very specific topics. But we also invite other users or scientists in the team that have been already using Bioterrain and have been able to publish really important results in their analysis. We also are open in inviting you to sort of giving a sort of small sort of lecture on your specific usage and how you've used it for your specific case study. So that's also open for this sort of Exploration Club. Okay. So I'll just give a few minutes, and I think my colleagues will gather that feedback from the team. Okay, if there's any questions that are sort of important for your data, or they're private to your specific case and you don't want to share it on the chat, you can always reach out to the support@bioterrain.com and ask us how to sort of get access to the system. If you don't already have sort of a license for the system, we can give you access for a trial period. Also for curating data, please send us a list of any data sets that you want us to add into the system that are publicly available, or any other questions that you're facing when you're exploring the system that you need further guidance in. Okay. So I guess we'll just give a few seconds as we get all those servers completed. And once that, this sort of concludes this weekly session for today. Okay. So thank you everyone who has already sort of finalized the survey, and thank you all the scientists who have joined. And I hope this was sort of a useful case study so you can use it for further analysis on the biotrain system.